Hi, we are from Group 3. My name is Lin Jiaxin with Bsb3513 and he is my partner, Yang Wenxue with Bsb22030.
Hello everyone, we will talk about T-cell activation and effector function in this video. There are about 30 to 37 trillions of cells in our body, and there are approximately around 20 to 40% of whole white blood cells population are lymphocyte. Lymphocytes are a type of white blood cell that play a crucial role in the body's immune system, helping to fight infections and diseases. Lymphocytes goes into three main types: B cell, T cell, and lastly, natural killer cell, also known as NK cell. And one of the facts that is unfamiliar, 70 to 80% of lymphocytes in our bloodstreams are made up of T cells. This also shows how important T cells are in our bloodstreams. Now, moving to introduction to T cells.
Hi, we are T lymphocytes, or you can call us T cells. We are a type of white blood cell. We are known as naive T cell, as we haven't encountered any antigens yet, so we are still not activated. Our job is to protect the host body from infection and maybe cancer. We were born in bone marrow, but we grow up in thymus until matured. Then we will relocate to lymphatic system and bloodstreams. We work well in adaptive immune system. When the immune system recognizes the threat, a specific T cell will recruit T cell to fight, and T cell will copy itself and making more T cell to defeat the intruders. These cells known as effector T cell.
This is CD4 T cell or helper T cell. CD4 T cell don't kill infected cells, but it will send signals to other immune cells to activate them. This is CD8 T cell or cytotoxic T cell. It will recognize the infected cell and directly kill the cells. Now, let's move to T cell activation. There are three signals for activating the T cells. First, antigen recognition. Nucleated cell are those expressing MHC, class 1 molecules, presenting antigen to T cell. For example, dendritic cell. CD8 T cell with T cell receptor, or TCR, will bind to MHC1 molecule on nucleated cell. For CD, for T cell, it will bind to MHC class 2 molecule, where you can find it on the surface of antigen-presenting cell, APCs, such as macrophage and dendritic cell. Next, signal 2, co-stimulation. CD4 T cell bind to APCs through receptor known as CD28-2B7, which is on the surface of APCs. While for CD8 T cell, it also have receptor CD28, and it will bind to B7 on nucleated cell. Why T cells have to co-stimulation? T cells require co-stimulation because simply recognizing an antigen is not enough to activate them fully. Second, signals ensures that T cells are properly activated by microbial antigens, not harmless foreign substances. Moving on, signal 3, cytokine priming. APCs secreting cytokine to CD8 T cell, enhancing T cell activation. Cytokines, a protein that used for cell signaling, such as interleukin or interferon. After 3 signals of T cell activation, T cell starts to proliferate and differentiate. This is naive T cell, remain unactivated and immatured, will then activated by 3 signals, differentiate into CD4 T cell and CD8 T cell. CD4 T cell and CD8 T cell will then undergoes proliferation, duplicating many of themselves. Both of them will differentiate into different cells. For CD4 T cell, it will differentiate into Th1, Th2, Th17, and TFH cell. Whereas, for CD8 T cell, it will differentiate into cytotoxic T cell and memory CD8 plus T cell. So right now, we will go through the function of each effector T cell. So what is effector cell? Effector T cells are specialized immune cells that actively mediate immune responses. They are capable of carrying out specific functions.
Hi, I am T helper 1 cell, or you can call me Th1 cell. I am specifically against the intracellular pathogens such as viruses and bacteria. I will be activated by interleukin 12 and start producing interferon gamma and activating macrophage to engulf pathogens.
Hi, I am T helper to cell, or TH to cell. I am particularly against helminths and parasites. I will produce interleukin 4, 5 and 13 to activate B cell for producing antibodies against helminths and parasites.
Hi, I am T helper 17 cell, or TH17 cell. I am particularly against extracellular bacteria and fungi. I will produce interleukin 17 and 22, which mediate inflammation and immune responses, which can promote neutrophils activation and recruitment.
Hi, I am T follicular helper cell, TFH cell. I am particularly against extracellular pathogens. I am able to promote B cell activation, promoting memory B cell development, and even antibodies production, such as immunoglobulin E or IgE.
Hi, I am cytotoxic T cell. I am important for eliminating infected and malignant cells. I can recognize the infected cell by receptor called TCR to bind to antigen that are presented by MHC1 molecules on the surface of infected cell.
Hi, I am memory CD8 T cell. I am important for long-lasting immunity, particularly against intracellular pathogens and tumors. I am able to respond rapidly to re-exposure to same antigen, providing a more potent and efficient immune response, and I can live about 30 to 160 days.
Well, that's all for this video. Thank you, and see you next time.